And as my title says, I'm here at the University of Sheffield in the School of Biosciences in plant photosynthesis in soil. And I'm going to talk about environmental regulation of stomatal development. And in this, I'm really going to be talking a bit about my own work and my lab, but also giving a bit of an overview of the field, just so you have a flavour for this. So hopefully as plant scientists, many of you are familiar with what stomata are, but just in case, they're these small pores in the surface of the leaf. And obviously they're there to allow the uptake of CO2 for photosynthesis. But of course, as an offshoot of this, you end up losing water, or the leaf ends up losing water. And this evapotranspiration is really important for two main reasons. Firstly, it's this process that actually drives water uptake and movement through the plant. So actually evapotranspiration is really required if you're going to have water movement through the plant. But the second thing is that it brings into question this concept of water use and water use efficiency, because obviously what a plant needs to be able to do is to maintain a functional water balance, one that allows it to continue with its basic biochemical processes, its developmental processes, without being inhibited. And as plant scientists, we need to be thinking about how plants use water. Now let's give an example. We always like to give numbers about here, but taking maize or corn, depending upon where you're from, an acre of corn will use something like 11 to 15,000 litres of water a day during the growing season. So, okay, that's quite a lot. Now, let's think about how much corn is grown across the US. And that's around about 92 million acres of corn. So if you do the maths, that's a phenomenal amount of water that's required for the irrigation of corn in the US across a year. And really encapsulates the issue with agriculture. We utilize a phenomenal amount of our freshwater resources for agriculture. And the problem is those resources are finite. So whatever we can do to understand how plants utilize water and how they can improve their water use efficiency. And here we're talking about the amount of carbon that they can gain for the amount of water that they lose. And so we really want to improve crop water use efficiency. And part of this involves understanding how stomata regulate this process. So when we think about stomata and gas exchange, primarily most people will be familiar with the idea that stomata can open and close in response to their environmental conditions. And yes, that is indeed one of the principal ways that stomata regulate plant gas exchange and water use. But the one I want to focus on today is actually the developmental side. So how development is plastic in response to the environment and how the environmental conditions therefore modulate the number of stomata that develop on a leaf. And this is just showing it very simply here, but we know, for example, that certain conditions result in an increase in the number of stomata that develop on the leaf surface, whereas other conditions lead to a decrease. And today, just for time purposes, I'm going to focus on light and how light is involved in the regulation of stomatal development. And why would plants respond to the light environment? Well, again, hopefully as plant scientists, we appreciate that the light environment is dynamic. So, it can change across the day, of course. So you can have periods of low light and periods of high light. It changes across seasons and it changes upon your local environment as well. So for example, plants within a canopy will experience different light qualities and quantities compared to those at the upper part of the canopy. And we'll probably hear a lot more about some of these things in Gabriella's talk, actually. So the light environment is dynamic and plants respond to this. Now I'm going to be talking about stomatal development, and I'm afraid this rather busy slide is my attempt to try and summarize some of the main points about stomatal development to allow you to think about some of the things I'll say later on. And this really summarizes a lot of, about 20 years or so of research in principally in Arabidopsis. So this is a lot of the works in Arabidopsis, but some of the stuff we do know applies to crops as well, although crops do occasionally do things slightly differently. But yeah, much of what I'll be talking about today is Arabidopsis. And if you were to look at a young developing Arabidopsis leaf, and what you would see is a variety of different cell types. And many of these represent cell types within the stomatal lineage. So if we look at this, and I've drawn it in a sort of linear fashion here, we can see that a cell can enter the stomatal lineage via an asymmetric division. And so you get this smaller meristomoid and a larger stomatal lineage ground cell. Now, a few things can happen at this point in time, but eventually one of the things that happens is that a meristomoid will transition to form a guard mother cell. And then, again, a guard mother cell may go on to form a pair of guard cells by this symmetrical division. And the trouble about showing it like this is that it sort of suggests that this is a linear process, that it's not very plastic at all, and it's defined. And that really isn't the case. You have decisions that are happening at all of these points in time that mean that you may continue along the stomatal lineage, continue along that trajectory, or the cell may exit the stomatal lineage. And so you may no longer actually end up producing a guard cell. And again, the other side of this, as you can see here, another important point to make, certainly in Arabidopsis, is that this isn't synchronous either. So these decisions are happening at various points within the leaf surface. Now, these different steps, these formative steps in stomatal development, are driven by specific transcription factors. And these are transcription factors in the basic helix family. So we have speechless here, which drives this asymmetric division, this entry into the stomatal lineage. You then have mute, which is required for the next step from meristomoid to guard mother cell. And then finally, pharma, which drives this last differentiation step. But work from Kaiketori's lab demonstrated that they're not doing this by themselves. They work as dimers. And in many instances, they're dimerizing with another set of transcription factors. And the principal one is ICE1, or this is also known as screen, and it's close homolog screen 2 as well. So these dimerize with these basic helix transcription factors to regulate these steps. So that's one level. But then let's just focus up here in this box, and we can see that also stomatal development is regulated by a receptor complex at the cell surface. And here you have members of the erector family of receptor, leucine receptor-like kinases, and they form a complex with too many mouths, which is a leucine rich repeat receptor-like protein, and circs as well. And they perceive these extracellular cysteine-rich epidermal patterning factor peptides. And I'm just showing you here EPF1 and EPF2 and stomagen. And we'll come back to stomagen in the middle of the talk. And what binding of these peptides to the receptor complex determines is the activity of a MAP kinase cascade. And this MAP kinase cascade ultimately regulates the activity or the stability of these core transcription factors. And principally, we know about the regulation of speechless and an ICE1 via this MAP kinase pathway. And in the case of speechless, generally speaking, this is negative regulation. So we tend to think that the MAP kinase pathway negatively regulates stomatal development, but we do know that there are certain points where actually it has the opposite role. And I've left out lots of things here, but really the take-home message is to focus on these early transcription factors, speechless and mute in particular, and we'll come back to the epidermal patterning factors as well. So I started in this field in around about 2007 when I moved to Professor Alistair Hetherington's group in Bristol. And at this point, it was known for example, that light regulated stomatal development. It was known that this was the case, and it was known that light positively regulated stomatal development, and we can show this quite simply here. So in this experiment, what we've done is we've grown plants to maturity, Arabidopsis plants to maturity, and in either a low light condition, which is 50 micromoles per meter squared per second of white light, or a high light condition of 250. And I should say that generally we grow Arabidopsis around about 130 to 150, and neither of these light conditions are stressful. You get perfectly healthy plants. It's nothing like some of the highlight work that's done to look at, for example, Ross generation. And so what you can do is grow these plants, take the mature leaves, make impressions with dental resin, and then count stomata. And I have, and my group has counted an awful lot of stomata over the years. And what you see is if you just count the number of stomata, it's very clear that when you grow plants under high light, you have a higher stomatal density on the leaves. So there are more stomata per unit area. However, what you can also do is count all the cells in the epidermis and then determine what the proportion of cells are actually stomata. And this is this stomatal index. And we tend to focus on the stomatal index because what the stomatal index is telling you really is a probability. When you have a division event, what's the likelihood that that is going to go on within the stomatal lineage and end up forming a stomate? And so we can see here that, light here is increasing the probability of cells going on to form stomates. So we tend to think it's positively regulating stomatal development. And I should say that's generally the case, but you have to be a little bit careful about when you're measuring index about this, because there are times when it can mean slightly other things, and it requires a bit of looking back at that stomatal developmental pathway and what the different factors do. But this was our starting point, and we knew light regulated stomatal development, so the question becomes how. And when you think about light and how plants respond to light, one thing that crops up is photoreceptors. It's not the only thing, but it's one of the things that crops up. You think about photoreceptors. And so photoreceptors are these molecules that are able to perceive and then initiate signaling processes that often ultimately end up changing either a physiological process or a developmental process. And in Arabidopsis and obviously many plant species, you have different categories of photoreceptors. You have the red and far red light perceiving phytochromes, and you have the blue light perceiving phototropins and cryptochromes. So to start off with, we thought, let's try and discriminate between these. So we'll do some experiments in monochromatic light. And so we grew plants in different irradiances, so different amounts of either red light, which would focus on the phytochromes, or blue light, which would principally focus on the phototropins and the cryptochromes. However, there is an issue here in that phytochromes actually can respond to blue light as well. So the blue light is not actually that discriminatory. But what we found is that in both red light and blue light, if you supply more red and blue light, you get an increase in the stomatal index. And so it immediately made us think, well, okay, it could be the cryptochromes or the phototropins, but we know it's likely to be the phytochromes. So we focused on the phytochromes. And to cut a long story short, we did a genetic analysis. So we looked at mutants in the phytochrome family. And there are 5 phytochrome genes in Arabidopsis. I'm only showing the results for 3 here because this is the Col0 ecotype, but we did this in other ecotypes as well with other mutants. And the upshot is that really in white light conditions, we principally only saw an effect when we mutated phytochrome B. So this was generally under high light conditions. We didn't really see much of a difference under low light. There was a little bit, but not much. So it was an attenuated response to high light that we saw a defect in phytochrome B genes. So they had a lower stomatal index. So it's suggesting that you require phytochrome B activity to positively regulate stomatal development in response to higher light. And now I'm going to flip forward and really summarize a lot of stuff, because the question that happens after this is, well, okay, here we have the photoreceptor, but we know that there's lots of stuff that happens downstream of the photoreceptors. So how are the photoreceptors affecting this stomatal developmental pathway? And so for the sake of time, I'm going to summarize a lot of this in just one slide. So this came about from a couple of groups' work, and it was shown that initially it was found that COP1 was found, it was found that COP1 was found to be involved in this process. So this came from Hong Kong Yen's group. However, they weren't able to identify the factor, and this came a couple of years ago when it was shown that COP1 is able to target ICE1 for degradation. So I should say that COP1 is an E3 ubiquitin ligase, and it was known because it had been found in light signaling screens to target light-regulated transcription factors for degradation by the proteasome. And here it was targeting ICE1 for degradation. So this seems to be a good means for photoreceptors to regulate stomatal development because the phytochromes and the cryptochromes regulate the activity of COP1. But there was some other work that was coming out around about the same time, and this was coming from Phil Wigg and Dominique Bergman. And they were able to show that PIF4, which stands for phytochrome interacting factor 4, so this is a transcription factor that is negatively regulated by the phytochromes. They're able to show this PIF4 binds to the species promoter and inhibits species transcription under higher temperature conditions. Now, the fact is, phytochromes are known to both perceive light and also be temperature sensors. And it's not hard to make the connection, therefore, that if they're doing this in response to temperature, they may well be doing it in response to light as well. And in fact, we had shown that PIF4 was involved in regulating stomatal development prior to this, but had not made this mechanistic link. So here we have two ways where photoreceptors, and I should say the cryptochromes are involved as well, this again came from another study, can regulate this stomatal developmental pathway. But is it the only way? And I'd say the answer is probably not. I'm going to give you a couple of more examples from the field where we're looking at this question of how light is regulating stomatal development. And the first of these is going to focus on stomagen. So stomagen. So this is one of these epidermal patterning factor like genes. So these are these cysteine-rich secreted peptides that are perceived by the receptor complex at the cell surface that regulates the activity of the MAP kinase pathway that ultimately then regulates transcription factor activity. And stomagen is unusual in this family because unlike the other members that affect stomatal development, EPF1 and EPF2, stomagen is a positive regulator of stomatal development. And we can see this here. We can see that in this stomagen RNAi line, you see significant reduction in the number of stomata that form, whereas when you overexpress, you get lots of stomata forming and you get this clustering phenotype, which often happens when you overexpress factors that positively regulate stomatal development. Now that's interesting in its own right, that stomagen positively regulates stomatal development. But what was further interest, and actually, the major point about this was that actually stomagen isn't expressed in the epidermis. It appears to be expressed in the internal tissue, in the mesophyll. So we can see this here. And this immediately led to the hypothesis that here we have the mesophyll. This is a tissue that's associated with photosynthesis, with light. So does this therefore mean that stomagen is the connection between light and stomatal development, between photosynthesis and stomatal development? And this is something that my lab and also the lab of Bunsen lab started to look at. And now I'm going to tell you something that, where there's a little bit of controversy in the field, or at least let's just say results that are different. So both my lab and the lab of Bunsen lab had been looking at the stomagen gene, looking at the promoter and looking at factors that can regulate stomagen. And we both found that within the stomagen promoter were binding sites for a well-known light-regulated transcription factor called Hi5. And so having this chip data available, saying high-5 binds the stomagen promoter, immediately you start to think, well, okay, we know that high-5 is regulated by photoreceptors. So again, this is a great link between photoreceptors and light regulation of stomatal development. And I'm just showing some of our data. What we did was we looked and we found via dual luciferase experiments where you overexpress high-5 in protoplasts and also introduce a construct where the luciferase report is driven by the stomagen promoter. We could see that high-5 could indeed regulate the expression of a transgene from the stomagen promoter. We looked at where high-5 is expressed, and it's expressed in the epidermis. So here we have a high-5 CFP fusion. It's expressed in the epidermis, but more importantly, it's expressed in the mesophyll as well. So it's where stomagen is. Everything is looking very good. And as we already knew from the literature, high-5 is light regulated, and here you can see its expression goes up when you move to high light from low light, and it goes down when you go from high light to low light. And this would map what happens when you look at stomatal development. Stomatal development goes up in high light, goes down in low light. So everything was looking great. And this is where then the results from my lab and the results from Ons and Lau's lab start to diverge. So having got this data, we looked at genetics. We looked at the mutants in this pathway. And we had the high-5 mutant. We also had mutations in a close homologue of high-5 called HYH. And we had the double mutant. And we looked at all of these under our low and high light conditions. And the upshot is that in these conditions, we saw no difference between these mutants and the wild type. So there's the wild type under highlight, and there's essentially high-5 HYH and the high-5 HYH double mutant. So we didn't see any difference. And we've got 5B in here just to show you that under these conditions, 5B still has a reduced stomatal index. So this was a bit disappointing. And we then looked at gene expression data, and we did light transfers with seedlings. So we grow seedlings under either low light conditions and transfer them to high light, or under high light conditions and transfer them to low light. And under these conditions, if you do this with a wild type plant, you will see, for example, the expression of stomagen goes up when you move to high light, and it goes down when you move to low light. And this is a wild type plant. And speechless goes up when you move to high light, mute goes up when you go to high light. And again, both of them go down when you move from high light to low light. And high-5, the high-5 HYH double mutant was essentially responding very similar to the wild type. So our conclusion is we couldn't find any in vivo evidence to support a role for high-5 regulating stomagen and hence positively regulating stomatal development. We certainly could find evidence to support the potential, but we couldn't actually find hard evidence to say it actually happened. And that's where our results and Ons and Lau's groups differ. So I think the best thing to say is at the moment, this could be due to growth conditions that we use. This may explain some of the differences between our groups and Ons and Lau's groups, but it leaves it a bit of an unknown as to whether high-5 is actually involved. So we're still at this point where we can certainly say that, you know, we have a module here that can explain what's happening with COP1 and ICE1 regulated by the phytochromes and cryptochromes, and PIF4 regulating speechless. Is that it? Well, here's where there's a grand story. You don't try and fix something that isn't broken. We decided to do some more experiments and got some results we were a bit surprised at. So I've just mentioned these QRT-PCR experiments, so expression analysis experiments, where we look at the expression of stomatal regulators after doing light transfers. And we've done lots of work where we've done low light to high light. But we started to look in the opposite direction, what happens when you move from high light to low light. And this is just focusing on phytochrome B, but these results apply to cryptochrome 1 and 2 mutants, and they apply to a 5e cryptochrome 1, cryptochrome 2 triple mutant. So we see the same thing in all of these mutant backgrounds. And that is that if you go from low light to high light, You will see the upregulation of speechless and mute. So just focus on those. They're the main regulators, these early steps. You always see that in a wild-type plant, whereas the photoreceptor mutants are blind to this increase in light. And so you're not seeing it, for example, in a 5e mutant here. But if you do the opposite experiment and go from highlight to low light, now the photoreceptor mutants do respond. And they're responding essentially the same as a wild-type plant does. So there's something different about going from low light to highlight. It's not the same, it's not the reverse process of going from highlight to low light. So these is not the same as, like climbing up a set of steps and just going back down the steps. Something is different is happening in the mechanisms here. And we wanted to investigate this. Now in Sheffield, there's a strong photosynthetic community. And so this got us thinking about photosynthesis and in particular, the light reactions of photosynthesis. And so here I've just given a schematic of the photosystems in the chloroplast, photosystem 2 and photosystem 1, and the electron transport chain. And in highlight, what happens is light is primarily pumping photons into PS2 and you get charge separation. And so you get electrons that are then passed on to plastiquinone. And this reduces plastiquinone and it then transfers it to the cytochrome B6F complex, plastocyanin, and onto PS1, which then eventually will reduce NADP to NADPH. And that's what's happening in highlights. You're getting lots of photons coming in and lots of electrons passing through. So you end up reducing the plastiquinone pool. But in low light, what tends to happen is that with fewer photons coming in and fewer electrons being generated, you end up with the plastiquinone pool becoming more oxidized. And this doesn't just happen if you have a reduction in light. You'll notice here that the photosystems, they have reaction centers that essentially have peak absorption wavelengths. So PS2 primarily absorbs in the red and the blue, whereas PS1 here in the far red. And if you focus on PS1 and illuminate with far red, what you actually do is oxidize the plastiquinone pool because you don't have electrons being released from PS2. So you get cyclic electron transport, but you're basically not replenishing your electrons to the plastiquinone pool, so it becomes oxidized. And actually, the plastiquinone pool is quite a good indicator of environmental conditions. So if you look at the redox status, it's quite a good indication of whether you've got highlight, low light, or enrichment in far red. And the good thing about this system is that you can use inhibitors to investigate it. And I'm just showing you here, DCMU is a specific inhibitor that blocks electrons from moving from PS2 to plastiquinone. So you can have a highlight condition, but by using DCMU, you block the electrons passing onto the plastiquinone pool so it will become oxidized. And that's the same as if you irradiate PS1 with far red light. So, okay, DCMU treatment sort of has connotations of low light. So what happens if you treat plants with DCMU? And this is what we did. And here's some results. And I should say I can only tell you a small part of this story, but this is going to be hopefully published, released next week in Current Biology. So the full story will be out there. But we treated plants with a non-lethal dose of DCMU. So 3-day-old seedlings we treated with DCMU and then looked at stomatal development as a time course. And what we found is that DCMU treatment inhibited stomatal development, just as we would expect growth at low light to do. If we did gene expression analysis of plants that were grown at high light and then treated with DCMU, 6 hours, which is typically when we start to see robust changes, we saw robust reductions in expression of speechless and mute. And this is mimicking to many, in many cases, what we see when we move from high light to low light. Okay, this is using an inhibitor, and there are issues with using inhibitors, although I should say in our defense, this is something that's been widely used in the hormone field. You know, hormones have been widely investigated using the hormones themselves or inhibitors. So Let's not beat about too much on the use of inhibitors. But we wanted to do it in another way, in a more physiological manner as well. So we wanted to use far red light to target PS1 and oxidize the plastic chrome pool that way. But here you have a bit of a problem, because far red light will also inactivate phytochromes, and we know that phytochromes are involved in regulating stomatal development. So we had to get around this issue, and the way we did this was, we were very lucky. John Clark Ligarius was very kind enough to provide us with a null mutant in the phytochrome, so all 5 phytochromes are knocked out. And so we did this experiment. We grew plants under highlight conditions, and then gave them a pulse of far red light and looked to see what happened to the expression of key stomatal regulators. And here we can see in a wild type Landsberg erector, we see downregulation of speechless, we see downregulation of mute. And when you do this in this final mutant, you see the same thing. So the effect of far red light here is independent of the phytochromes, and is still leading to downregulation of stomatal development. And so our conclusion from this is, along with a lot of other work that we'll see in the current biology paper, is that the redox status of the plastic chrome pool is involved in regulating stomatal development. And so that leads to really this conclusion that light regulation of stomatal development occurs at many points in the pathway, and there are many opportunities to modulate stomatal development within the leaf. And that, yes, it involves classical photoreceptors and photoreceptor pathways targeting these key components such as ICE1 and speechless. But we also have signals coming out of the chloroplast in response to changes in the redox status of this electron transport chain that are also affecting the activity of this pathway. So I hope that gives you a good overview of what's going on here, but obviously there are other environmental factors as well, such as CO2, humidity, etc. And these are all things that you have to consider when thinking about environmental regulation. And with that, I would just like to thank the people who've done the work. In particular, we have Nick Zulius, who has worked incredibly hard on these projects, and Nick and Jim Rowe, who's now in Cambridge, and he did a lot of the imaging work, and Jordan, who did a lot of counting, and Magda, who did a lot of gene expression work, and obviously many of my collaborators who this work wouldn't be possible without them, and of course funders from the BBSSC, Newton, Leave Human Moral Society. Thank you very much.
Excellent, thank you, Stuart, and congratulations of the new paper coming out. That's really interesting stuff. Look forward to reading more about it. Okay, so I'll encourage people to ask questions in the Q&A, or put your virtual hand up, and we can ask Stuart some questions. So let me kick off though, Stuart. We have a couple already come in, but let me kick off. As you said just at the end there's so many factors which influence stomatal development, and you show that highlight causes an increase in numbers, but is that completely dependent on the drought condition and the water availability as well? There's no point having more stomata if you haven't got any water there as well.
Yes, no, you're absolutely right. I mean, the experiments we do are in well-watered conditions, and it's already been shown, for example, that abscisic acid, which is classically associated with drought responses, tends to negatively regulate not only stomatal aperture, but also early decisions in the stomatal lineage. So yeah, you always have these pathways that are essentially at war with each other, and part and parcel of it is essentially this balance. Where does the balance end? And so this is part of the flexibility of the pathway, is that you can have something that would allow a process to happen, but then it's not terminal. It's not as though you've made that decision. There's no going back. So certainly you could enter the stomatal lineage, and then if it became droughted, you could terminate those decisions.
Yeah, it's important, I think, when you do these experiments, not to say that your conclusions is that's it, that's the most important thing in development. It depends on the it's very situational.
No, absolutely. I mean, you can think about all, the temperature will have an effect, yeah, and the humidity, all of these things, yeah.
But the fact remains that there is some biology going on that you've.
Yes, absolutely, yeah.
Okay, so we have a first question from Javitha, and they ask, do any of the transcription factors involved in retrograde signaling determine stomatal development?
Well, I mean, I've already talked about one here today, Hi5, which is classically, obviously, also involved in retrograde signaling. And again, I mean, I just have to say that one possibility is that the different experimental conditions that we use compared to other labs might explain why we didn't see anything and other labs did. So there is the potential, but admittedly, I can assure you that the Hi5 was looked at quite intensively for the project with regards to stomagen, and also with regards to the one involving oxidation of the plastic quinone pool, and I can say that we didn't really see any strong evidence there. In terms of others, we've had some tantalizing evidence over the years, but many things don't often essentially turn into substantial, what I would say, results that I would be confident in saying, yes, this definitely has effects. So, you know, there are things like ABI4, there's a possibility, but nothing that I'm absolutely, I would hang my hat and say, yes, this is involved. But there's lots of, the trouble with, yeah, there's lots of retrograde signals, and it's pinning down which might well be involved.
Okay, so I see there's a hand up, but we've got a couple of questions on the Q&A first of all, so I'll come to you, Tracy, in a minute. So for a question from Alonso Mateo. He asks, is there, well, it says very interesting talk first of all, but is there a relation between the expression of stomagen and the PQ pool?
Good question. Good question. So we do have some evidence in the paper that will be released. So again, I think this boils down to the concept that one direction doesn't, the response in one direction doesn't mean that the response in the other direction is just going down the same route and backwards. And I think this is something that we've seen several times. So for example, our evidence with regards to this oxidation of the plastic quinone pool with regards to, for example, a high light to low light, actually, we didn't see much evidence that stomagen is involved. But I wouldn't discount that it's not involved in the opposite process. But my point is that they don't have to be just like, you know, one is doing it by increasing stomagen and hence it has to be via downregulation the other way. No, I think that they can be independent, and we do have some evidence that if you reduce the plastic quinone pool, that yes, you do see changes in stomagen expression. And also there's evidence that, for example, stomagen responds to reactive oxygen species as well. So this has been found in the root, strangely enough. And so there's a possibility there as well.
Okay, so I'm going to bring in Tracy. There we go, Tracy. If you can, you're unmuted now, so I think you should be able to.
Can you hear me?
Yes, indeed we can.
Oh, great talk, Stuart. It was really, really interesting, really clear. I have a question about the plastic quinone pool and how does it work in terms of, like, you said you sort of did that for the 6 hour. Does it have to stay in then reduced to oxidized states for that length of time, or is there some sort of integration of the signal? And could that also be a determinant of different numbers in a sort of linear way or something like that? How do you think that works? Because that's fascinating.
No, you're absolutely right. That is a fantastic question. And it's at the heart of something that, you know, really we would like to get to the bottom of. And it's something that as a field, it's a difficult question to answer. How are these things being integrated in time? And when do you reach a critical threshold? And it's the way that I'm sort of thinking at the moment that, I mean, if you think about, you know, developmental decisions and the cell cycle, you know, often in plants, it's been found it's around about 24 hours for the cell cycle, there or thereabouts, and I'm sure someone can correct me, but it means you've got a window where a developmental decision can happen. But that window might be condensed by, for example, the photoperiod, and we've got good evidence to suggest that, you know, this is the case. And so there are so many things going on, and one of the things that I didn't show is you know, a lot of work is focused on speechless, because it's the entry point, so it's a natural focus. And you know, the stomatal community has found there's a number of post translational modifications of speechless that either affect its stability or its activity. And so if you think you have a pool of speechless within a cell, and you're having all of these signals ping in, then perhaps it is the accumulation of these within a certain window, within the photoperiod, that ultimately leads to a decision process. And that would be sort of my take, that you probably have a threshold that is achieved at some point during the photoperiod that will determine whether something will progress or whether it won't progress, or it might even just be held in stasis for a while. And I think that's what I would say about this, because, you know, we both know that like environment's dynamic, and that you are going to have days. I'm looking out my window now at a very dull day, so we're going to have days when it's more likely that the plastic window pool is oxidized for long periods, whereas for others it's going to be fluctuating throughout the day. And I think, yes, how these dynamic signals feed in this asynchronous environment is it's really a fascinating one to answer. And I'm sorry, I'm not sure I have.
It'll be super interesting to look at it, though. It's
No,
really,
that's right.
great. Thanks very much.
Thanks, Tracy. Okay, so I mean, we've got still a few questions coming in. Obviously, they're stimulating quite a lot of thought here, but I'm going to just take one more question so we can move on to Gabriella. So final question from Scott Hayes. It's got a bit more specific. So is it possible the reduced stomagen induction in the 5B mutant on low light to high light transition, could actually be down to a reduced chloroplast development in this mutant?
Hi, Scott. Good to hear from you. No, there is that possibility. I mean, this is where it becomes very difficult sometimes, because unpicking one thing from another is always difficult. And anyone in the photoreceptor community will know that 5B does regulate chlorophyll biosynthesis. So, you know, the plants look naturally paler. So you're absolutely right. I wouldn't discount that there is a connection there.
Okay, so I'm going to ask you, Stuart, so hopefully you can see the Q&A there. There's a question from Luke Bell in the Q&A. And then there's a couple of questions in the chat as well from Ashley and Murray, which I'm not sure other people will be able to see. But if you can get in there and you can answer those by text, that'd be great. Because I'm gonna move on now.
Okay.
But thank you very